hard to fit it into 12 minutes.
How did you figure out that compound was related to cilia? How did they ever first?
People, ever since, Van Leeuwenhoek invented the microscope, people have been rather interested in looking at things underneath it. And at some point along the way, someone just kind of took a bit of pond water, looked underneath, and saw things swimming about. And I'm sure that they said, I wonder why they move, and I wonder how they move. And with various stains and such, they probably were able to identify cilia or flagella or various other mechanisms by which these creatures, if you will, can move about.
Just as a non-scientist, if I were looking at them, I see, and maybe it's easier for a non-scientist, if I see cells, one of them has thin hair-like structures to move fluid in your lungs, the other has thin hair-like structures to swim around in fluids. It makes sense to start looking for similarities. What's really shocking is how similar they are.
Yeah.
When Tom was showing cross-sections of the human cilium and cross-sections of the chlamydomonas, you can't tell them apart.
Yeah.
It's really amazing.
It is extraordinary how conserved cilia and flagella are. The flagella that you see in this chlamydomonas, which Phil described as a plant, actually it doesn't know what it is. It's part plant, part animal. It moves like an animal, but it's chlorophyll like a plant. So it's kind of right at that evolutionary divergence, if you will. But the proteins, the structure, the genes that encode proteins that make up a flagella is virtually identical to every protein, every gene that makes up a human cilium. They may be put together slightly differently, but nevertheless, they all still work the same way, which just tells you how important these structures are. I mean, if this is something that has gone through evolution or creative, intelligent design, whatever you want to call it, that this has been sustained for billions of years. It just says this is kind of something unique. So.
Does this have implications for other diseases other than respiratory? Is that, or
Right.
you're not worried about that at this point?
Well, being a lung doctor, I'm never worried. No, I'm just joking. I think the implications here, and this is what I'm hoping we'll be able to get across, is it's very difficult to do drug studies in human airway epithelial cells or the cells that line the brain or the cells that line the retina, because when you grow them in culture, they don't seem to act right. They don't act as they do when they're in the body, unless you do something that is, perhaps special, to get them to do that. But then you can't look at a lot of them at the same time. You can't do mass studies. Chlamydomonas, on the other hand, you can grow, 1 billion of these cells with the exact same defect that has been fully characterized, that you can then potentially test drugs on. You can see whether or not you can correct the function of the cilia in terms of its movement. And that would have implications for respiratory disease, ear disease, sinus disease, male infertility. But the other thing about chlamydomonas is that beating flagellum also has sensory functions. It functions an awful lot like the monocilium that you see in the kidney and the biliary tract. And in fact, many of the genes that were originally described that lead to primary cellular dyskinesia were confirmed in chlamydomonas looking in the flagellum. So you could also make a similar sort of argument if you have a way of detecting the ability of the chlamydomonas to sense its environment, that if you can correct that with a drug, that if you use a drug and you can correct that so it moves away from light, which you typically do, that could be perhaps another tool. But our focus has been primarily on how do you help these kids who have primary cellular dyskinesia or chronic ear disease or persistent sinusitis or recurrent bronchitis. And I'll let Alan tell you how clever that was, but I thought it was a pretty interesting idea. So, oh, yes, ma'am.
How did it occur to you to get together with a mechanical engineer? I mean, how did this come about?
Oh, it's. Do you want to tell the story? Or?
I have one version.
And so I get 5 minutes for rebuttal.
I have one version. I was in an administrative meeting with Susan Dutcher, who's a geneticist on the topic. And Susan, who really, by all rights, should be here because she's really part of the important part of the mix. Susan knew Tom and knew the problem Tom was working on, and she ran into me at an administrative meeting and was telling me about cilia, and I was telling her about my interests in oscillations and basically things that move back and forth in a number of ways. I'm a vibrations person, and I've looked at the heartbeat and other problems. So Susan Dutcher actually, I think, put it together.
And I cannot rebut that. He's exactly right, because Susan and I were actually sitting around talking about, okay, we really want to kind of pursue this, both at a genetic level and a clinical level, and I was just whining, I suppose, saying, gosh, it would be really useful if we had someone who could help us measure cilia function and help us understand how cilia move. And she says, I think I may know someone. And it must have been around the same time as the meeting. And what's also been rather striking, first and foremost, this would never happen without the CDI. This would never have happened. I mean, I would never have met Phil, and Susan and I probably wouldn't have been talking about a project if it wasn't for the fact that there was the possibility that we could actually fund this sort of, harebrained scheme that we have. But
Pun intended, right?
no pun intended. But the other aspect of this was that once we got people engaged and we saw what we could do and we saw where this is potentially going, then we started to get people even outside of our field. So it's, Phil has been taking the lead for biomedical and aeronautical engineering, but we also have who else? Mechanical and aeronautical engineering. We have biomedical engineers now involved. We have obviously Steve with internal medicine, other internists who are involved in this project. We are talking with people, other specialists who are thinking about how the kidney works, how the biliary tract works, how the bile ducts work. And it's just kind of remarkable how this is expanded. So, yes, sir.
Where are you on the CDI grant timeline? And do you have hope of NIH funding or anything because of the seed money?
Yes, as a matter of fact. So we just finished the second year. We're now at the beginning of the third year of a 3-year project. In May, we responded to a request for applications to the NIH in an engineering, interestingly enough, a biomedical engineering institute that was submitted, and we actually plan to submit another award more to one of the lung institutes, the National Heart, Lung, and Blood Institute, in February. Because I think we're going to have the preliminary data regarding the genetics of this Amish community. We're going to have all of the fantastic data regarding modeling human disease and chlamydomonas and all the fine work that Phil has done. I'm probably going to shoot myself in the foot by saying I think they'll like it, but I think they'll like it.
I also should mention, and Tom, we submitted a patent disclosure on a screening method to look at the effects of pharmaceuticals on chlamydomonas. So it's not for sure whether, it's new to us what happens with patent disclosures and whether this is something that we'll actually file a patent on. But it was an interesting idea. There were some interesting tricks that we're going to try and do to see how good the swimmers are and how they respond to drugs. But that's an interesting thing. I'm not sure how many of the CDI projects will end up in patent disclosures.
I'll guarantee you it was not on our plans, our to-do list when we first submitted the project. Are you telling us we're done? I'm sorry, we're done.